And actually, again, with every speaker, we like to hear something about your personal side that we wouldn't otherwise find on the web.
Okay, thank you very much, John, for the introduction. Something private, not on social media sites. When I was young, and I'm probably giving my age away here, I was an avid science fiction reader and used to play with computers. Now, hands up those of you who know what a ZX Spectrum 81 is. There are a few of you. Okay, so I bolted together the ZX 81, I worked with a Spectrum, but why that was important for me was my father was an accountant. And he used to sit, I remember when I was a nipper, to use an expression, when I was young, he used to sit at the big dining room table with his big sheet of paper, a pencil, and a slide rule, and he used to do the accounts for the company he worked for. Then one day, he couldn't understand, why am I playing with these computers? Why did I ever go to the newsagent, buy a magazine, type in the code, save it on a cassette recorder, if you remember that? And then one day I showed him VisiCalc, which was a program on a Spectrum, which was a spreadsheet, which allowed him to do the accounts. His eyes just popped out of his head. And can you imagine now finance and controlling without spreadsheets or without computers? Well, it was then. So that was something that was brought home to me very early, how technological change can just move whole environments. Right, to my talk. Working together to solve the translational conundrum. What I want to do in the space of the next 20 or so minutes is provide you with some evidence that the pharma industry, working together with academia, needs to open itself up in order to be able to face the challenges of the future. I'm going to, pardon me, I'm just going to give you a little bit of theory at the start about what open innovation is, what the different definitions are, and then I'm going to go into some concrete examples that are very new, which I want to show you the way we have worked together with organisations such as Innocentive, BioMedX, and the SGC, in order to be able to work on that challenge. Also, point of full disclaiming here, not only do I represent my organisation, Boehringer Ingelheim, but we're also a member of the Structural Genomics Consortium, and I sit on the board and have the pleasure of interacting with Al on a regular basis and the other scientists in academia and industry. So now I have to steal the slides. Next slide. Start off with that number, $2.6 billion. There's a paper just been published by DiMasi. Came out a few weeks ago, online version, which is telling us that the average cost of taking a drug from the start to launch in R&D costs is $2.6 billion US. That has increased over the period of time. DiMasi and the Tufts Centre for Drug Development has looked at that regularly for several years. That cost has gone up by over 8% above the rate of inflation. At the same time, the productivity over decades has been going down, and if you divide the one by the other, you can see we've got a problem, because that sort of investment can't be sustained. So we have to find another way of doing things. Why is that happening? We're facing squeezed margins, increasing costs, competition from drugs that are no longer covered by patents. We're also facing a challenge in the research world. Increasing information, better understand biological complexity of targeted disease is the key. Many of the failures that are happening in the clinic is because we don't understand the biology enough. A conventional closed model of innovation is unsuitable for some of our medical needs. You've heard some examples from Al a few moments ago in rare diseases and in tropical diseases. So in response to the limitations of the closed model, a growing number of organisations have come together over the past 2 decades to share ideas, and I can read it down here, with each other and with the wider community. And what I want to do is walk you through some of that today. But before I do that, I'd like to pause. There was a question earlier on in the discussion about the software industry, and I think the pharma industry, the biotech industry, can learn a lot from the steps in opening up the process of software that the software industry has gone through. If you look at the classical closed analog innovation, traditional innovation in an analog world before the internet, it was centralized, it was top-down, loved by pharma executives everywhere. A call it collaboration hard. It still's collaboration hard. How long does it take us to get those bilateral agreements in place that we need? And I don't think it's scalable. Open or digital innovation, and I think this is characterized with the story of Linux. One person's approach to finding a new open version of a software system. 90% of the large computers run on Linux. All your Facebook, media, Twitter is run on servers which are running Linux. Your smartphones, if you don't have an Apple smartphone, but if you have the rest, they're running on Android, they're running on open source software. I believe pharma has a lot to learn from that approach. Now I want to make a pause. Theory, I said, to talk a little bit about definitions. Open innovation, we know the term. It's first coined by Henry Chesbrough in 2003, I apologise, to describe the concept that companies use external ideas as well as internal ones to advance their R&D. Actually, he was looking at a very limited form of open innovation, how companies could go beyond and form bilateral collaboration with other parts of the pharma biotech ecosystem. You can also see, and I was ashamed, Jens, I don't know whether you're still here today, this morning you didn't mention the Google Ngram Viewer. This is a lovely tool. You can spend hours sitting on the sofa on a notebook computer looking at the frequency of words in the English language between the 1900s and around about 2012, 2014. And you can look, for instance, at the frequency of the term open innovation. Okay, Henry Chesbrough was supposed to have coined that term in 2003. I guess someone beforehand must have been using that term, as you can see it here. It's going up. Actually, if you compare that with words like open data and open science, they're fairly flat. If I was to plot out open access, you wouldn't see the curve of open innovation, because open access is much higher. So what I want to do now is that open innovation is a general term, but actually you can dig down. You can talk about open source, which is pioneered by software development. Open source projects making the fruits of open collaboration free to anyone. Open data. The Open Data Institute in London describes open data as data that anyone can access, use and share. Open access, that relates to publications and periodicals. There's a major shake-up in the conventional publishing model. Literature is digital, online, free at the point of use, and usually free to reproduce in some form. Open science is a general term which relates to the practice of science the practice of sharing science that others can contribute. Lab notes are freely available. We saw an example earlier on from Al, and the research and its underlying methods can be shared. So I always urge you, when you're talking about open innovation, open source, open data, be clear about what exactly you mean by that. Next point is about selective revealing. And I was quite ignorant of this until a young student with a master's thesis came to me and explained the term. Basically, it relates to a paper by Alexei et al. And these authors talked about the necessity of revealing different things of your organisation in order to be able to achieve more. And basically, the two blocks are, you can either reveal your problem, or you can provide your solution. Revealing your problem is best demonstrated with crowdsourcing. You have to have the vision and the bravery to say what you don't know in order to be able to seek a solution for it outside. I'm not talking about sharing information within the scope of a bilateral disclosure agreement. I'm talking about no disclosure agreements, freely making that problem or that solution available to the outside world. And actually, when I move forward, I'm going to be focusing a little bit on path extension here, revealing either your problem or revealing a potential solution in the form of a chemical probe. Again, open innovation, as I want to intend it today, is not just related to bilateral collaborations between 2 or 3 different organisations. We're all trying to access the power of the scientific crowd, and therefore, for the rest of my talk, I'm going to be actually giving you some examples of crowdsourcing platforms and public-private partnerships which are trying to harness that crowd. I'll start with crowdsourcing. Crowdsourcing is the act of taking a job traditionally performed by a designated agent and outsourcing it to an unidentified and generally large group of people. That's the white paper version. As far as I can determine, that was coined in 2006 by Jeff Howe in Wired magazine. He also talked about his soundbite version, the application of open source principles to the field outside software. Again, making that link, what can we learn from the software industry and the move to open source and open software? I believe strongly, however, in order to be able to do that, you need a reliable framework. And what I want to do now is give you a couple of examples. One, BioMedX, an organisation based in Heidelberg. Christian Tidona, who's CEO, is sitting in the audience today. And Innocentive, a platform company which actually sprang out of Eli Lilly. What does it mean, crowdsourcing? Well, it means you have to formulate a challenge. Remember selective revealing? You have to say what you don't know, and you have to say it in a way in which you will encourage sensible answers. Then you use an online platform, as presented by Innocentive, in which you pitch your problem to the external world. Solvers have to register themselves on the site and provide you an answer. And if you want to use that answer, you have to be able to reward them. The nice thing about it is it's all set up. You work with a broker who does that for you, but you have to learn how you formulate your question and, most importantly, apply the necessary resources in order to be able to evaluate those answers that are being provided with. Then you have to reward the ones you want to use. On the right, I won't go into great detail, but you see we now actually, with the latest iteration, with our 12th call in this area, specific scientific questions which we're asking to help our own research. We've had 1,500 registrations worldwide from 8 challenges. We haven't got the data for the remaining 4 challenges yet. We received 280 solutions, and I can tell you they're all over the world. It's not just your East Coast big institutions of the US which are providing you solutions. These solutions can come from anywhere. Also interesting to see with the timing of them. Many of the good solutions come just before the deadline. Scientists are the same everywhere. 19 awards we've given. We've rewarded the people who provide us with solutions monetarily with 19 awards. And we're actually working on 17 potential collaborations. This is the problem. Remember I said open to closed, closed to collaboration hard. Here's your challenge. You're trying to set up individual bilateral collaborations with lawyers and tech transfer officers, and that takes time, unfortunately. What have we learned from our Innocentive challenges? Innocentive is a process, an infrastructure which works. The scientific question, we need have had practice in how we specify that challenge in order to focus the answers that we're getting. We also need to have the appropriate resources available in order to be able to review and analyse the solutions. It's also important to reach out to the right solver community. It's not just one big community which you can always access, but there are different nuances to it where you go in order to be able to address that. But it really is a novel way to find new ideas and collaborators. And it actually has given us a positive external recognition. We do the same sort of thing with BioMedX, however with one important more than subtle difference. So we use a crowdsourcing platform of BioMedX in order to be able to formulate our challenge, but here we're not just looking for solutions, we're looking for people with solutions. And then we go something that Christian has set up, which is really great to participate in, is the so-called boot camp, where Christian and his team, together with scientists from a particular organisation, really puts the young scientists through the mangle. Sleep deprivation, hard work, and they have to work through their ideas, present ideas, and they have to work in teams, very important. Big difference perhaps between some of the things that go on in academia and what happens in the pharma industry is we're very dependent on projects which work in teams, so people have to be able to get on together, they have to work together, and sometimes they have to put their individual pride back a little bit in order for the success of the team. Detailed project work and plan is set up, and then we set them up. We've got a pre-agreed agreement, so we don't have to go away afterwards and start negotiating tech transfer offices. It's a set. We spend the time in defining the project and the scientists involved, and then we set them up and they get to work. And then we have the option to say, if we like what we get, we'll reward them and we'll take it on board ourselves. So this for me is extremely important because one of the challenges we faced with Innocentive is the crowdsourcing approach is good, but you need to have a proper infrastructure in order to be able to convert your creative idea into a potential product. And that really for me is the essence of innovation. You can see the team on the right. The first team's been working now for a few months in Heidelberg. Culturally diverse, smart, bright people putting together, working on epigenetic regulation and the development and progression of COPD, something which is very important to us in the organisation. 399 proposals, full project proposals were 170, internally evaluated 99. Remember, I said you have to have the resources, you can't just run this and hope that it'll look after itself, you have to invest the time to do it. And we invited 23 of those people to the boot camp. They all enjoyed the boot camp, but they were pretty damn tired at the end of it, I think I can say. Just going to change tack now, and actually I can shorten my presentation by one slide because Al was so kind enough to demonstrate the challenge of the distribution of different proteins and how the publication citation index is seen. But this, I think, just to reinforce that message, because I believe that is a relationship that holds true for a whole range of different protein families, is the so-called streetlight effect. I'm looking for my quarter I dropped, he says. Did you drop it here? No, I dropped it 2 blocks down the street. Then why are you looking for it here? Because the light is better here, the streetlight effect. And this is what we have to guard against. If we shine a light on a series of proteins where everyone's publishing on, we may not be looking in the right place for our new target protein that we need in order to be able to address a disease. Al showed you examples of the kinases. There are examples here, more specific examples on the upper part of the slide. Bromodomains here, the distribution of publications and the patents, and then what the genetic information, short hairpin RNAs, GWAS studies and OMIM, and this holds true for protein methyltransferases too. So the whole field of epigenetics is one which is also seems to be suffering from the very similar challenges we saw in the kinases or the nuclear hormone receptors, that academia is focusing on certain proteins and getting good publications at it. The genetic information is telling us we should actually be looking elsewhere. So, the model of the SGC. John, you asked a question earlier about why is patenting important? I think what Al was talking about, there are many diseases in which it's probably not good or not for a pharmaceutical company to make that big investment necessary. It needs to be able to recoup that big investment. So I don't think patents are dead, but I think there is a space in the early stage, which is demonstrated the way we work with the SGC, is that we need to be open. Because freedom to operate is more important than cramping everyone with certain patents. At some point you need to have a drug discovery project. So the way we work with the SGC in our organisation is that we work in the public-private partnership. And we work together with a public-private partnership in order to be able to use the expertise of the academics in protein structure. We work together with our medicinal chemistry expertise in order to be able to define a series of chemical probes. These chemical probes are what we hope biologists out there can use to interrogate their systems. I've heard also systems biology. Well, systems biology is only good if you have the tools in which you can perturb that system so you can generate and test your hypotheses. Our idea behind that within the SGC is the academic crowd, remember the term crowd and crowdsourcing, then uses those, but the implicit agreement or tacit agreement you have is that there's no patent rights, that you provide those probes from industry unencumbered, to use the legalese, and allow and encourage the academics to publish around it. At some point, however, once you've got a better understanding of the biology, then it's time to run your drug discovery project. That's a costly business going through the different phases of preclinical and clinical development, and an organisation such as our own or any other organisation, pharma, needs to be able to recoup its investment. But I think there's a fundamental difference. There's an area where, on the right-hand side, you're looking at what I say, industry, the proprietary area, but we need to understand the biology better, we need to perturb the systems, and this, for me, is where we should have a creative commons area. Draw your attention to the community-driven wiki resource, which we published last year, the SGC, devoting a site, chemicalprobes.org, as a one-stop shop for people to go to in order to be able to find information about what chemical probes are available in particular areas. Good probes are very useful to the academic world, bad probes can mislead them. And the whole idea behind this is let's put our heads together and make sure that we define good probes, we define the criteria for those good chemical probes, and then we encourage their use. One example, in the last 5 minutes of my talk, I'm going to be talking about the mammalian switch sucrose non-fermentable complex, better known as the BAF or SWISNF. This is a responsible protein complex which is part of the epigenetic control. It controls how genes are expressed. Remember, your genes, your DNA is wrapped around histones, which is folded into your chromosomes, but the way it is methylated and acetylated determines how those genes are expressed. One of the complexes which governs gene expression is the BAF complex. And there's actually a bromodomain on it, bromodomain 9, which is part of that. And we know from the literature, here we go for how you start a drug discovery experiment. The BAF protein complex is required for positioning of the nucleosomes on the DNA. We know that components of the BAF complex are mutated in 20% of human cancers. Good starting point for maybe something which could be interesting for us as a drug discovery project. Bromodomain 9, this one here, is required for survival of mouse leukemia cells, and we know that this gene is amplified and expressed in 18% of lung cancers. So we set up a drug discovery project, classical pharma, and we started to look at it. And actually, we have now published that. Today is the 26th of May. It's out today in the Journal of MedChem. I'll come to that in a minute. This is the molecule. We've made the structure available. We used a structure-based screening approach, orthogonal screening. We used all the techniques that we have in our organisation in Vienna and in Biberach in order to be able to find a molecule which is able to bind to and influence the bromodomain 9 protein. It inhibits the bromodomain 9. It was generated by structure-guided design. It has a high affinity for the site, key element in defining it as a chemical probe too. It's selective and cell permeable, and what's really nice about this, and this is perhaps something which was important to not to underestimate in the JQ1 story, which Al spoke a few moments ago, is that it also acts in vivo. It has pharmacokinetic properties, which allows you to be able to use it in disease-related animal models. Then we tested it in a disease-related animal model. We used a disseminated mouse leukemia model. You inject cancer cells which have been transfected with a tag which allows you to fluoresce, to be able to track them. On the upper part, you see the mice in the control group. On the lower group, you see the mice which have been treated with our chemical probe 9564. Actually, the results are quite disappointing. We expected more. The effect on survival was only 2 days as well. This actually led us to we don't really understand properly what's going on, and maybe our hypothesis is not right. So what we did is we're offering this chemical probe out to the scientific environment via the SGC site. We actually have this on the front page of JMedChem, published out today. There's the molecule, there's the complex protein, there's the structure. We published it, it's freely available. We also got a very nice Viewpoint article highlighting the importance of this molecule in being able to understand the biology. It's also available on the SGC website. Anybody can go there, unencumbered, call it down, see the characteristics of that molecule and be able to get hold of it and test it. The material transfer agreement is the standard one that you have with the SGC, and all we encourage them is people tell us and publish their findings with this molecule. It's only we have taken that step that GSK did with the SGC several years ago in making that probe freely available. We have our own. We've learned from the BRD4 story. So this is publications according to the different bromodomains for overall bromodomains for the 9. You can see the JQ1 publication. You can see when the JQ1 was synthesized and published by the SGC. We have our own BRD4 program in clinical development at the moment. We have profited from that study, and in fact some of those studies which Al mentioned about acute myeloid leukemia were done in association with our scientists at the Institute for Molecular Pathology in Vienna. And here, because we believe so strongly in that, we're making the 9564 available. I'd like to end my talk, hopefully by trying to summarise that I believe in the pharma industry we need to learn the lessons of the software industry in that we approach our research and development. We have to be much more open. We have to be prepared to reveal things to the outside world, what we know and what we don't know, and we need to work with the academic world in order to be able to provide the tools which allows us to interrogate the biology. Remember, the biology is key to understanding the disease mechanisms and hopefully being able to make better drugs. Finally, I'd like to draw your attention to the fact that we as an organisation have sponsored a special edition of Nature. It came out on the 12th of May. I was hopefully going to have a few examples with me, but they haven't reached us in Ingelheim yet. This entire supplement is built around the concept of open innovation. There are a wide choice of articles, some of actually the information I've used in my talk today. I would highlight also at this point that we had no editorial control whatsoever over the content, but nevertheless I can highly recommend the content. We have also, because we've sponsored it, made that edition of Nature supplement available. Anyone, even without a Nature subscription, thank you, can go and access that and call it down and read those articles, and I urge you to do so. And on that note, ladies and gentlemen, I'd like to finish my talk. Thank you very much.